Okay, you all can see the screen, everyone?
Yes, sir.
Perfect. So let's start with a quick recap of the last class. We discussed some fundamentals of DNA replication. So anyone who would like to quickly talk about it in 3-4 minutes? Rabia, how about you? Since you're the only one speaking, I guess Parker is not able to speak or something. Either you or Iram. So what about you, Rabia?
So I don't have my book with me right now.
No worries. You can see the screen that I'm sharing. You can just take the reference from the screen and speak what we discussed if you want. Otherwise, Iram, what about you? Can you do?
Yes, I can.
Yeah, so let me ask Rabia first. Rabia, would you like to go ahead and try with things on screen, or would you like Iram to do it today? Are you there, Rabia? Okay. Iram, if you can hear me, you go ahead, please.
Yes, sir.
Yeah.
So in the last class, we started with replication of DNA. Double helical structure of DNA was given by Watson and Crick in the year 1953. And by this, they also hypothesized a mechanism by which DNA replicates. First, the DNA unzips itself by breaking the hydrogen bond, and then those two strands form complementary strands. So one strand is called as the template strand or the parental strand, and the other strand is the new strand. This is called
Right.
the semi-conservative model of DNA replication.
Right.
Then later, Michelson and Stahl in 1958 proved the semi-conservative nature of DNA. They used E. coli as their model organism, and they used E. coli because it is a prokaryote, which means it has a single thread of DNA. Then they used ammonium chloride having a heavy nitrogen N15 isotope to culture E. coli bacteria. In the parental generation, the cultured bacteria which act as a parent having N15. Then for the first generation, we introduced the parent with normal nitrogen. After 20 minutes, we see that
Yeah, you see, are there, Iram?
yes, sorry, there was some disturbance.
Yeah. It's okay.
Yeah. For the first generation, we introduced the parent with normal nitrogen. After 20 minutes, we see that there is a hybrid DNA. Then in the second generation, after 40 minutes, we got two strands which were light and the other two strands which were hybrid. And this further proved that DNA has a semi-conservative nature.
Right. Perfect. So this was basically what we discussed in the last class majorly. So any doubts with respect to Michelson and Stahl's experiment? No doubts?
So
Yes. Yeah.
So I have a doubt. So this what we discussed is at molecular level, right? But so what is the difference in molecular level and chromosomal level?
Yes. So what we are discussing here is the DNA, like the DNA molecule, right? It's a good question. What you ask. What is the difference between DNA replicating at a molecular level and a chromosomal level? So basically, from the last class, we know that DNA is nothing but chromosome is nothing but a very, very condensed form of a very long DNA polymer, right? So normally, when does the cell needs its DNA to be replicated completely? DNA replication means making two copies. And that only happens when the cell is about to divide, right? You will be, I don't know if you people know this, but most of the cells in our body have lost their ability to divide further. They are terminally differentiated cells, whether they are the cells in our brain, which are neurons, or the cells in our skin, or the red blood cells, or tissues, most of the tissues, muscles, they do not divide further. They are terminally differentiated. When they die, something called stem cells then form new cells that replace it, or when an injury happens, or in very specific parts of the brain, these neuronal stem cells can give rise to new neurons. So overall, in a multicellular organism's body, not all the cells are dividing actively. But there is one type of a cell, one cell type, which actively keeps dividing, and those are the germ cells, right? The precursor germ cells. In reproduction, we have studied, right? Gametogenesis. So gametogenesis and oogenesis constantly keep happening in every sexually reproducing organism. So there, when the DNA is supercoiled into chromosomes, before replication, that chromosome becomes loose, okay? It unwinds. And after unwinding only, the machinery that we are going to study in today's class that is responsible for replicating a DNA can reach that long thread of DNA and replicate it. Also, what we do not often pay attention to in schools, or even teachers don't pay attention to when they're teaching, is the fact that DNA is a very, very long molecule. So for such a long molecule, you cannot decide to start at one end and replicate it like a zipper. Like you start a zip from one end and keep zipping it and replicate the whole DNA in one go. That doesn't happen. So mostly there are origin of replications, we call. So there are multiple origin of replication in a long DNA, which makes one chromosome. When it opens, simultaneous replication starts from multiple places, and then all these join together to form a new DNA molecule and a new chromosome. So the chromosome number becomes double. Do you understand? So this is how it happens at the chromosomal level. So first the chromosome has to become a little loose and open itself, and wherever it will open and DNA will be exposed, okay? Just like you open a thread and then you make a copy of that thread from the spool. Remember I gave you the analogy of how a long thread is kept in our homes in the form of a spool, right? Does
Yes,
that make sense? Yeah. So the
yes.
spool opens different like sections of the thread, becomes loose, and then molecules reach in between and start replication from multiple sides. Okay. So that's how it happens at the chromosomal level. Ultimately, the full length of the DNA has to be replicated. And you can imagine it's a very, very long molecule, and it has billions of base pair, correct? In the whole body.
Yes.
Yeah, so all these millions and billions of base pairs needs to be replicated. So the speed at which these enzymes work is phenomenal. You will be surprised to know that, do you know the enzyme that is responsible for DNA replication? We are going to start from that only today. So let's just start that part, okay? Is your question answered,
Yes.
Okay, great. Before I go forward, I just wanted to tell you that so whatever Messelson and Stahl did, they did it on E. coli, which is a prokaryote, right? Now, we know that a prokaryote and a eukaryote cell has a lot of differences. So obviously, a very fundamental question will be asked that what we see in a bacteria, will it also hold true in a multicellular organism like a plant or an animal? Now, In animals, it is a little tricky to do that experiment for various reasons. But there was a group that was headed by a scientist called Taylor. So Dr. Taylor and colleagues, they basically repeated the same experiment that Messelson and Stahl did, but it's very similar experimental setup, but instead of using a heavy something, they used a radioactive thymidine. Okay, so they used a radioactive thymidine. Okay, now you know that we have ATGC, right? So the T, if it is radio labeled, then when you will make new strand, if you get the signal only from half of the strand and the half of the strand does not have that signal, you can again prove that it is a semi-conservative method, right, of replication. So Dr. Taylor used radioactive thymidine, and he detected the distribution of newly synthesized DNA in the chromosome. Okay, and the model system that he used was a plant. It's called faba beans. Anyone of you know about faba beans? It's a bean. Its scientific name is Vicia faba. Faba beans are very, very interesting also from a point of a different aspect. Many people have when they eat faba beans, they show allergic responses, but it's a plant and it is eukaryotic system. So when they could show the similar kind of results in a eukaryotic system, and they also did it, by the way, just after Messelson and Stahl released their results in 1958 only. And this experiment also proved that DNA in chromosomes of all model organisms, whether they are prokaryotes or eukaryotes, they replicate in a semi-conservative method. Okay, so you can write down that Dr. Taylor and his colleagues Dr. Taylor and his colleagues proved that DNA in the chromosomes of eukaryotes, DNA in the chromosomes of eukaryotes also replicate in a semi-conservative manner. So remember this name because this is not very important for your boards, but might come as a question in your entrances, because NCERT also talks about Dr. Taylor and colleagues, but they do not explain the experiment in detail. So don't worry about the experiment. And I think you can understand that what they did is they used faba beans, and right from the seeds, when they allowed them to grow, genetically they tagged everything that's going to form, new DNA that's going to form and cell that's going to divide by using radioactive thymidine. Okay, so it was a little complex experiment, but it proved that the DNA replication method of the semi-conservative method of DNA replication is true for eukaryotic and complex systems as well, not just for a prokaryote, which has a single DNA. Okay. So that is also kind of an answer to your question, Iram, that how it happens in chromosomes. So chromosomes loosen up, DNA exposed, then the machinery comes, replicates it, and then it's stitched together. Okay. And write down another point that they used radioactive thymidine. That's the difference. They used radioactive thymidine to detect the distribution of newly synthesized strand. To detect the distribution of newly synthesized strand. Okay. And the model system that they used was a plant called faba beans. You can just Google and see the image. I think you'll all understand and relate to it. Okay, everyone. Is that clear to everyone? Shall we move forward?
Yes, sir.
Okay, great. So let's come to this point, and we will be talking about the machinery or the DNA replication machinery, as it's called. The DNA replication machinery, including the enzymes. Remember, any chemical reaction inside a cell happens because of enzymes, right? Enzymes are called biocatalysts. The fundamental similarity is the same, just like in a chemical reaction, when you use a catalyst. What does a catalyst do in a chemical reaction? You must have used catalysts in your chemistry experiments in school, right? Anyone would like to tell me, what does a catalyst do? What is the role of a catalyst in chemistry reactions?
Rate of reaction.
It decreases or increases?
It increases.
Increases the rate of reaction. And how does it do that? How does it increase the rate of reaction?
Substrate formation.
Something I'm hearing some words being mumbled by Iram, but those are not clear. Come on.
Substrate formation.
Substrate formation. Would you like to explain it a little? What do you mean by substrate formation?
No, I forgot. Like we studied in lab. I forgot.
You studied when?
In biomolecule.
Oh, you studied I think biomolecules is a chapter which is shared both in chemistry and biology. Is it?
No, I'm talking about the 11th, which is later.
For 11th biology, you have biomolecules. Yeah, correct, right?
Yeah.
So you must be. So anyone else would like to give a try before we discuss Iram's point? What does a catalyst do? I'm not even asking about, if you know about enzymes, great. But as I told you that enzymes are just biocatalysts. So if you would like to tell me just about catalysts, that's also fine. Someone else was speaking, I think Parker or Rabia. Come on, try at least. At the worst, you will be wrong, which is completely okay. So this is true that, okay, I got something in the chat. Rabia says it speeds up the chemical reaction, which is same as saying it increases the rate of reaction. Rate is anything over time. So speed is distance over time. So same thing, right? So you're saying the same thing again, Rabia, but would you like to just add something to it? Lowers the temperature, Mariam says. Lowers the temperature of what? So if I put a catalyst in a beaker with some reagents and try to heat it up, it will not allow it to heat up. When you say it lowers the temperature, you also have to tell the context. Lowers the temperature of what? So Rabia says now there is a discussion going on. Rabia says it lowers the temperature or pressure needed to start the reaction, and that's correct. So catalysts, what they do, they lowers the activation energy in terms of temperature, or whether she's right, if it's pressure, temperature, or any other kind of a condition that is required. So enzymes make the reaction happen. Sorry, catalysts make the reaction happen at a low temperature than it is required to happen at. So normally, if you need some chemical reaction to happen, let's say at 200 degrees Celsius, but a catalyst, if you add a catalyst, it can allow it to happen at 40 or 50 degrees Celsius. So it lowers the temperature which is required to start the energy. Okay, because most of the time that temperature is needed to provide kinetic energy to the molecules, correct? So that they collide and the chances of collision increases, and then chances of chemical reaction also increases. And also this energy is required for formation of the bonds and breaking of the bonds. Now, enzyme provides a substrate sometimes, right? What Iram was trying to go to point out here. So she was right. Sometimes it provides a substrate where A and B, which are called reactants, can come and form bonds with each other and get released as a product. So you must have studied A plus B gives C with an arrow, and on the arrow you write catalyst or an enzyme, right? That's what we do for biological reactions as well. Is that clear, everyone?
Yes, sir.
Okay, so now that you know about catalysts, what do you think is a biocatalyst? Simple. A biocatalyst is a catalyst that is helping in biological reactions. Okay, and enzymes, we give them the name enzymes. So in our body, if you look carefully, the same chemical reactions are happening inside a living cell that you go and do in a beaker. Okay. For example, let me ask you a very simple question. If I want you to produce heat energy in your life, in your chemistry lab, how will you do that? Iram, you have to
Increase the
produce heat.
temperature.
How? Through a reaction, tell me. Don't say I'll turn on the heater and it will take electricity. I'm talking in terms of reactions, chemical reactions. Do you know of any chemical reaction that produces heat energy? How did early cavemen used to produce heat during winters? I know today we can turn on the heater, but when heaters were not there, what did human used to do to produce heat? They used to gather wood and then they used to rub rocks, create spark, and burn anything that can be burnt. So basically they were not knowing, but they were causing a reaction called combustion reaction. Have you not studied combustion reaction, people?
Yes, study.
What is a combustion reaction? Can anyone tell me? Combustion reaction, simple. You burn anything. That's a kind of a combustion reaction, isn't it? Anything that is combustible, which means it can burn. And what is the reaction that happens when something is burning? Let's say you burn a paper. What happens? This is science. If you people think that you can alienate your life from science and still score good marks, that's not going to happen. You have to think. So scientific temperament was a movement started by our early leaders everywhere in the world. In India, it was started by our first prime minister. Many such people started this revolution on societal level, which is called scientific temperament. And it says that we as citizens, not as students of science or scientists or researchers or doctors, just as a citizen, a common citizen doing anything in his or her life, still we need to develop a scientific temperament, a sense of curiosity towards what's happening in our day-to-day lives. And you can learn a lot of science just from your day-to-day life if you're curious. So we all burn paper, or have burned paper. What happens? Mariam says release of CO2. That's half the answer. Where does that CO2 comes from, which is released? Quickly. Or I can just finish this weird looking diagram and move ahead. Parker says addition of oxygen. That's good. So when anything burns, it takes oxygen. A very, very classical experiment which was done way back when people did not used to know about much of science was that someone was burning a candle and then they kept a glass on top of the candle. And the candle stopped burning after some time. It got exhausted. It got extinguished. So what changed? The CO2 that, the O2, the oxygen that was available to the candle finished and the candle no longer can burn, right? Yes, the cutoff O2. We still use the same science in all our fire extinguishers. All the methods to extinguish a fire relies on this one logic. Do not allow the fire, cut the supply of oxygen to the fire and the fire will go down, right? Except for the part when you are putting water. So when you put water, you are lowering the temperature. So fire needs a higher temperature. When you put cold water, fire also gets extinguished. That's one way. But in sometimes you cannot use water, especially in electrical fire or something. Then you use a fire extinguisher, which has either foam. Foam makes a layer or a gas. All these makes a layer between fire and the atmosphere, like a blanket. You can also put a blanket on a fire and can just extinguish the fire. So it's a simple logic. Fire or combustion needs oxygen to happen. It's a chemical reaction. So if I ask you to produce heat in your day-to-day life, you will take some wood or paper. You can even take your cookies that you are eating and you burn it. You burn anything which has carbon in it, which sugars do have. So carbohydrates have a lot of carbon. Our body is made up of carbon. Cookies are made up of carbon. Dried chips are made up of, everything is made up of carbon only. When you burn it, everything organic, I'm saying. When you burn it, oxygen is taken and the carbon becomes carbon dioxide and that is released. Okay. So if I take a cookie and set it on fire, it will do the same reaction that will happen if I eat that cookie. If I eat a cookie, it will go into my body. All the carbohydrates will be absorbed. That carbohydrate will go in each and every cell and we call it glucose, C6H12O6. That C6H12O6 will go inside the cell. Then we will breathe in oxygen through lungs. That oxygen will also go inside the cell. and that oxygen will be used to break down that glucose. So basically you are doing the same reaction, and then you release CO2 out along with heat energy. That's how our body functions. Do you understand, everyone? Did you ever think that you are actually doing something equivalent to burning a cookie inside your body? So that's exactly what you are doing. But the question here is, when you burn a cookie outside, what temperature does it reach? What temperature does it need? You have to keep it on a flame. Let's say you use the lighter or something, a source of fire, and you put a cookie on it. It will take some time, some temperature it will reach before it burns and start producing CO2. Am I correct? Yeah. And it's very
Yes.
high temperature
Yes,
at which it burns. Even when a paper burns, it reaches a very high temperature. But when the same glucose is being oxidized inside our cells, what temperature does it happen at? 37 degrees Celsius. Can you burn a paper at 37 degrees Celsius outside the body? No. But you can burn carbohydrate inside the body. When we say burning calories, that term is actually not a misnomer. It's actually equivalent. We do burn calories, but we burn it at 37 degrees Celsius because we have enzymes in our body that help us to do the same reaction at a very, very low temperature, at our own body temperature. So that's science. Okay. So enzymes are very important for each and every chemical reaction that's happening inside the body. Now let's come back to this point. The diagram that you see in front of you is, it shows the machinery and enzymes which takes place in a DNA replication. Now let's start with something fundamental. So look here, we will start from this side. So there is one strand which has a 3' to, let's say where does this strand go? I'm just following. So this is the strand to 5' polarity. Can everyone see this? We are seeing from this side, everyone.
Yes, sir.
Okay. The other strand is 5' to, it goes like this, it's opened and it's here, 5' to 3' polarity. So these are the two strands of the DNA. And you can see here, this is how our DNA is inside the cell. It is helical, right? The DNA is helical. So you need something to first unbound it, to make it straight like a ladder. That's where you can, so imagine a zipper. Is it easy to unzip or zip anything, your bag, purse, your jackets, when it is straight or when it is folded?
when it is straight.
Exactly. So same logic is here. You have to make this helical molecule, and this helicity, this shape, this helical shape I told you makes the DNA very, very stable. That's why it does not react very soon. So when you are making it go back and become straight, that's called unbounding or unwounding. You are making it now susceptible to reaction, so it becomes straight. So here it's not helical. Okay, it's straight. And to do that, one enzyme which is used, which comes and plays a role, is called topoisomerase. Okay, so topoisomerase, first write down. Topoisomerase is the enzyme that helps in unbinding the helical DNA into non-helical form. Okay, so it keeps going and it's like straighten it up. Once it's straightened, what do you see next? Do you see these purple things here? These things? Everyone see these things? They're on both the strands, right?
Yes, sir.
Remember I told you that a single strand is not very, very stable. But what's making it single in the first place? What's making it single is this enzyme called helicase. So it was a helix, which is opening. So topoisomerase can do both the things. It can bind and unbind both. Helicase opens. Can you see that it's opening the DNA? Like,
Yes, sir.
like an arrow, it's going and opening the zipper. It actually looks like a zipper. If you just make a thing like this, and you will think that you are just unzipping a zipper, right? So it unzips. And when it unzips, the double-stranded helical structure becomes single-stranded, and now it's unstable. So you need these single-strand DNA binding proteins. So these proteins bind to this single strand and keep it stable. Okay, because it has to replicate. And when it will replicate, it needs every base which opened up to be at exactly the same place so that the new complementary base can be attached on the opposite strand exactly at the right place. So without these DNA binding proteins, single strands won't be stable. Okay. So these proteins come, make it stable. And now there is a fundamental problem here. Now the strand that got opened, one is called the leading strand and the other is called the lagging strand. Now, why one is called the leading and one is called the lagging strand is because the main enzyme that is responsible for making DNA, replicating DNA, is this enzyme that you see here. I'm just this block, these two blocks. Can you see this, everyone?
Yes, sir.
So when you are making new DNA, you are basically polymerizing. You are making polymer. So the enzyme, and remember most of the enzymes, when they end, their names end with ASE, ASE, topoisomerase, helicase, polymerase. So polymerase is the main enzyme that replicates DNA or that makes the complementary strand. Now this polymerase is a DNA polymerase because it's making DNA. And what this diagram does not tell you is that its full name is DNA dependent DNA polymerase. Why I'm calling it DNA dependent DNA polymerase? Because in biology, there is something called RNA dependent DNA polymerase as well. In reverse transcription, which retroviruses can do, we use RNA as a template and we make a DNA from it. Remember reverse transcription, everyone?
Yes.
So that polymerase will be called RNA dependent DNA polymerase. Here, the template is also a DNA, and we are also making another DNA strand. So it's called DNA dependent DNA polymerase because it uses DNA as a template to catalyze the polymerization of deoxyribonucleotides. So write down DNA dependent DNA polymerase uses DNA dependent DNA polymerase uses DNA as a template, one strand of DNA as a template, one strand of DNA as a template to catalyze the polymerization, of deoxynucleotides or deoxyribonucleotides. You can even write deoxynucleotides, it's fine. Deoxynucleotides. Okay. So nucleotides, I already have told you, are the monomers of DNA, correct? One nucleotide contains what? What are the three components of a single nucleotide? Anyone quickly? What is a nucleotide, people? We did it in the last. What are the three components of a nucleotide?
Phosphate, sugar,
Phosphate,
nitrogenous base.
sugar,
Nitrogenous base.
yes, and nitrogenous bases. Perfect. So these three has to be present to make a nucleotide. And what is a nucleoside? What is a nucleoside? How is it different or similar to a nucleotide? Remember that? You don't forget what we did in the last class. Otherwise, as someone is writing in the chat, but not speaking. Rabia is saying my voice is breaking. Is it breaking for others as well?
No, sir.
Okay, so my voice is not breaking for Iram, which means Rabia, it's your network. So Iram, what is nucleoside? How is it different from a nucleotide? Remember, I told you an easy way to remember it. What goes towards one side or towards inside is called nucleoside. So in a DNA, if you consider DNA as a ladder, the backbone is made up of phosphate and sugar. And what goes inside are the bases. So if you just take a base and a sugar, it's called a nucleoside. So nucleoside is nucleotide minus phosphate group. Is it clear, everyone?
Yes.
Yeah, perfect. So this is what DNA polymerase do, okay? Now, DNA polymerase has one problem in evolution. The problem is it can only polymerize in one direction. It only works in one direction, from 5' to 3' end, which means from a hydroxyl, from a what is at the 5' end, a phosphate or a hydroxyl group. Remember what was at the 5' end, people?
Phosphate.
How do we? Yes, the 5' is phosphate and the 3' is OH coming from the sugar, right? So the DNA polymerase enzyme can only work in one direction, which is 5' to 3'. So write down the DNA dependent DNA polymerase. Now we can also just write DNA polymerase or DNA pol. And also let me tell you, it's very specifically pol 2. So there are three kinds of polymerases: pol 1, pol 2, pol 3. Polymerase 1, polymerase 2, polymerase 3. So it's specifically DNA polymerase 2, and 2 is Roman. So you can write DNA pol 2. Okay. So this DNA pol 2 can only work in 5' to 3' direction. So if this is the direction on which it works, what should be the direction of the template strand that it will take as reference? It's not very difficult to understand. If polymerase, if you are running on a treadmill and you are running towards east, where should the treadmill be going towards? West, right? Perfect. So if DNA polymerase can make a long DNA single strand in 5' to 3' direction, what strand it should take as a reference? Look here. So this is a DNA pol. Again, if you can see, I'm now marking it with red here. And look that it is making a strand which is starting from 5' and going towards 3'. And the template strand that it is taking is 3' to 5' direction. Okay. So the strand which is 3' to 5', on that strand, you can very easily make a continuous long DNA strand, because that is the direction in which DNA polymerase works. So no problem. So that's called leading strand because it takes less time to be synthesized. It leads. The other one is called lagging strand because now you can understand the problem. Because the other template strand, so what I'm marking in blue here is template strand, right? 3' to 5'. Here the template strand is 5' to 3'. But 5' to 3' is also the direction of DNA replicating enzyme, which is DNA Pol 2. So what happens the moment you open the DNA, when it gets opened up, that opening is known as replication fork. It looks like a fork, right? A fork road like this. So at the DNA fork, this 5' to 3' template strand will have to wait. It will open, adequate amount of bases. Then let me use a different color, which is purple here. And you tell me on this strand, the leading strand, what is the direction of Pol activity? This, right? 5' to 3'. But on this strand, the polymerase works in this direction. Okay. So for this to happen, the strand has to first open, and whatever strand opened, it will make a strand. Let's say from here. Sorry, let me try to. So look here, everyone, carefully. You can see there is a break, right? So from here all the way to here, the DNA Pol 2 worked and made a fragment from here to here, let's say. But do you see this part? This is also a fragment, and here the work is still in progress. So this is a RNA primer because it's a single strand, and the enzyme is DNA primase, which keeps moving forward and keeps adding one nucleotide at a time. But you see there is a break here. Can you all see a break here?
Yes, sir.
This break is because DNA Pol started working when this helicase was here, when only this much DNA was opened. Now see this, now it has opened more DNA. From, let me erase this. From here to here, now it is open, right? So another polymerase will come and will sit here and will start making again in 5' to 3' direction. And again, helicase will open some more DNA. So for this strand, there is no problem. Polymerase can keep going as helicase is opening. But for the other strand, there is a problem. The polymerase have to bind, make a fragment, unbind, again wait for helicase to open, then it will go here, bind, make a fragment, and it will go here, bind, make a fragment. So on the lagging strand, the DNA replication happens in fragments. Is it clear, everyone?
Yes, sir.
Because of the directionality problem. And because it happens in fragments, you also need to stitch those fragments together. And that stitching is done by an enzyme called ligase. The word ligation means sticking together. Okay. So ligase just joins the two fragments on the lagging strand. Is that clear? Everyone?
Yes, sir.
So there is additional work that has to be done on lagging strand. Okay. Now, very simply, just to give you an analogy, let's say there is a two-lane highway one is constructing. Okay. So this is lane 1 and this is lane 2. But the rule is this is east side, this is west side facing. But the rule is the roller can only move from east to west. Okay. So for this lane 1, the roller can start here and keep making, and keep making. So there will be a vehicle in front which will keep putting all the debris and the roller will keep flattening it up. But on lane 2, there's a problem. The problem is, again, it can just move from east to west. But now, if the opening or the or the vehicle that was putting down sand, bricks and all is coming from west to east, then first, let's say if it put this much of sand, then roller will come here and go in this direction because roller has a direction fixed. And it will wait for it to put this much. Then again, roller will come here and go from east to west. Now again, the vehicle is going from west to east, but the roller will again come here, go from, you understand what I'm doing here? Only if the roller was allowed to go from west to east also, there would have been no problem on the opposite lane as well. But that's not the case with enzymes. Enzymes are molecules that have evolved over time, and how they function is based on how they interact with other molecules. So DNA polymerase has this limitation that it can only polymerize DNA from 5' to 3' direction. So the template strand, which is 3' to 5', becomes the leading strand, and there is a continuous DNA formed on it. Whereas the other template strand, which is 5' to 3', on that there is a lagging strand formed in fragments, and that fragment is called Okazaki fragment. Okazaki discovered that mechanism. Okay, everything is clear? What is Okazaki fragments? Okay, so write down. You wrote about the directionality of DNA-dependent DNA polymerase? People, yes, no? Are you there?
No.
Okay, so write down. The DNA-dependent DNA polymerase catalyzes polymerization reaction. The DNA-dependent DNA polymerase catalyzes the polymerization reaction only in one direction. Only in one direction, which also happens to be a very famous band, by the way. One Direction. Anyone of you hears or like One Direction?
Yes.
Yeah, that's great. So that's DNA polymerase for you. It only works in one direction. That is 5' to 3'. It was Eramosadius, right?
Yes.
Okay, cool. I also like One Direction. High five. So only in one direction, which is 5' to 3'. Next point. This creates some add-on complications. This creates some complications in the DNA replication process. Create some complications in the DNA replication process. As a consequence, one strand with one strand, one strand is replicated in a continuous manner. one strand is replicated in a continuous manner. And which will be this strand? In the bracket, write down the strand with the template of polarity 3' to 5'. Okay, so the strand which will have a template strand from the original DNA, which is 3' to 5', there it will be continuous because new strand will be 5' to 3', which is the direction of polymerase. Okay, done, written.
Yes.
Next point, whereas the other strand will be replicated in a discontinuous fashion. Whereas the other strand will be replicated in a discontinuous fashion. And which one will be this strand? The strand which will have a template 5' to 3', right? So in brackets again, write down the strand having the template with polarity 5' to 3'. See, the original strand which is coming from the parent DNA, that does not need to be synthesized again. That is already there. So it just need to be opened. So polymerase is not going to do anything on that strand. So that is a template. So all we have to see and be careful about is the template, which is the strand which is synthesizing, the newly synthesized one. And the new synthesis can happen continuously in 5' to 3' manner. So for that, the template strand has to be 3' to 5'. Okay. But if the template strand itself is 5' to 3', then you cannot make another new strand in the same orientation. Therefore, it becomes discontinuous. Okay. But remember that in any case, a new DNA strand is not formed in a 3' to 5' direction ever. DNA formation, DNA replication has only has one direction. That is 5' to 3'. So you understood the issue and the complexity, everyone? People, is it clear? Is there any confusion? Anyone who has confusion, just raise your hand or write in chat. If no confusion, then write down. This discontinuous strand is called Okazaki fragments. This discontinuous strand is called Okazaki fragments and is joined together by an enzyme called DNA ligase. So some enzymes and their functions you should know by heart. Polymerase, it catalyzes the polymerization reaction. Ligase, stitch up Okazaki fragments, ligates the Okazaki fragments. Helicase opens the DNA helix. Okay. These are the most common, most important ones. Okay, now one important thing here. I told you the whole story, but I didn't tell you the beginning. How does DNA polymerase, which is an enzyme, knows that now I have to start replicating the DNA? Right? So DNA polymerases cannot initiate the process of replication on their own, right? They're not autonomous. Like they can, otherwise these enzymes are always present in our cells. So what prevents them from replicating the DNA in a neuron? I told you that most of our cells in the body do not replicate the DNA. So what happens is replication cannot just randomly happen from any place in the DNA. As I told you, there are some defined, well-defined regions that they found even in the E. coli DNA, which E. coli just have a single DNA. Still, it has multiple regions in that sequence from where replication begins. And these regions are called origin of replication regions, ORI. Okay, in short, ORI, origin of replication. So write down the DNA polymerase cannot initiate replication randomly at any place. The DNA polymerase cannot initiate the replication randomly at any place in the DNA. Okay. The replication begins at specific sites or specific regions in the DNA known as origin of replication. Now, if a cell does not want to replicate its DNA, all it has to do is to keep the origin of replication covered, unexposed. Simple. So if your origin of replication is unexposed, DNA polymerase cannot bind there and cannot start replication. Does that make sense, everyone? Perfect. All this took, decades of research to figure out what we are studying in just one paragraph. So think about how beautiful the biological system is, okay? And how well regulated it is, but it doesn't mean it's foolproof. All our diseases, developmental defects, embryological defects, where we cannot replicate DNA properly. Trisomy. Now, can you understand how trisomies happen? How there can be one extra copy of a DNA during replication event, right? So biology is not foolproof. There are always mistakes that happen because it's a dynamic living system. But over time, if mistakes accumulate more than a limit, it will just kill the organism. So organism also have to survive. So every organism evolves new and new machineries and molecules inside its body. And this was the question that we are going to address in chapter 7 when we will study evolution. Evolution is the most misunderstood concept of science, I think. If not after, then as equal to the origin of universe. So we still do not have definite answers of an origin of universe, but we do have some very, very concrete answers in terms of proofs and evidences for origin of life and evolution of life as well. So when we'll study that, we will again come back to these questions and ask, how can a system work and evolve on its own? Why you do not need a perfectly designed system? So the whole point of science believing that you do not need a designer is because everything in biology is very, very flawed. So sometimes we think that our eyes are a beautiful system, right? There can nothing be as beautiful as a human eye. See, we can see from it. Don't you think so? Human eye is perfect, a design to see things. Anyone? What do you think? A human eye is a very perfect biological design, isn't it?
Yes.
It is not. It is a very flawed design that evolved and got selected in nature. It worked for us. So in computer science, there is a saying in coding and programming. When I started learning, I got to know that they say, if the program works, don't touch it. Okay. Even if you do not understand how it is working, if it is just working for you, let it work. So same is for biology. Human eye is not at all a perfect design. If I will teach you about human eye, I don't know if it's in 12th class. Oh, but in evolution, there will be a part where we will talk about octopus and human eye together. We'll do a comparative analysis. Yes. So I'll briefly try to tell you as much as I can. But a human eye is a very, very flawed design, and you will understand why. So if I would have asked you to design an eye, you would not have designed an eye like it is really in our body. Okay. There is a very simple way to design an eye, but our eye is just a flawed design. That's why we have a lot of issues that happen. We have a blind spot right there in sitting in our retina. You all must have labeled a human eye, right? You know about a blind spot? So there's a blind spot, and there are the ways the photoreceptors are arranged in retina. They're all very haphazard, but it works. It works, and it's selection of the fittest. And it, because of eyes, organisms survived, and they are surviving. Okay. So let's come back. We will ask these questions in evolution, but now let's understand the mechanism. The mechanism is more or less clear, right? Anyone has any doubt? People, any doubt with respect to this DNA replication machinery? The enzymes? Okay, if not, then let's move forward. Now also write down for your entrances, write down some facts. So in a eukaryotic cell, like a plant or a human cell, when does the replication of DNA takes place? I asked you a question, and I told you then just before a cell divides, and just before a cell division, the S phase, which is called the synthesis phase. The S phase is where the DNA replicates and become double. Okay. And yes, so write down DNA replicates during the S phase of the cell cycle in eukaryotes. And also, if this does not happen in a regulated manner, so there are many defects or issues that can come. And if it is not coordinated well, then you will end up making more DNA, but the cell won't divide, let's say. So that will lead to polyploidy. Remember I told you about polyploidy? Animals cannot tolerate polyploidy. So animal cells die when they are polyploids, but plant cells can tolerate polyploidy. But now do you understand why polyploidy happens? Because this replication begins, but the cell does not proceed into the cell cycle and does not divides. Is it clear, everyone?
Yes.
Yeah, great. Okay, so this is the major thing about replication. So the kind of questions in the next class, what I'm also planning is we will also do in the class some questions from previous years, entrances. We will try to solve some questions which come as statements. So which of the following statement is not true or correct about DNA replication? So there'll be 4 statements written, and all the 4 will be very, very similar to each other, just differing in 1 or 2 words. And that creates a lot of confusion sometimes. So we will try and do some of these questions in the next class. Okay. Okay, so we understood DNA replication. We understood when it happens. We understood how it happens, which is the most important enzyme, and what is the mechanism. Now, what after replication? What is the next in central dogma? Replication is from DNA to DNA, DNA dependent DNA polymerase. What next? We have to make, yes, we have to make a RNA from DNA, right? So that is called transcription. Now, one thing you will be good at now, without telling you anything, if I ask you, can you tell me which enzyme is responsible for transcription? Pay attention to what you are making, from what are you making, and what are you doing, the process. So can anyone tell me the name of the enzyme that does transcription? What are we making? RNA. How do we make RNA? Similar way we made DNA. Here, instead of deoxyribonucleotides, we will use ribonucleotides, right? AUGC, not ATGC. There's no thymine in RNA. So one thing is, again, we have to polymerize. It's a long molecule, so we have to make polymer of it. And the enzyme will be called RNA polymerase because we are making a RNA, isn't it? Correct?
Yes.
But we are making a RNA from what? From a DNA. So the enzyme's name will be DNA-dependent RNA polymerase. Was it difficult or was it easy to understand? So if you go to memorize these things, it will be more challenging. But if you understand, transcription is making RNA from DNA. Replication is making DNA from DNA. So DNA-dependent DNA polymerase. In transcription, the major enzyme is DNA-dependent RNA polymerase. If it was possible in nature to make RNA from a RNA, what would have been the enzyme called? RNA-dependent RNA polymerase, which does not exist. Like we do not know, like we have not figured it out, and it does not exist as of now. But it's possible that it may, something of that sort may have existed when life evolved, like millions of years ago. But then there was no need for it, so it did not get selected, and it's not anymore. But we do not have any RNA-dependent RNA polymerase because we cannot make RNA from RNA. Correct? So if they ask you which of the following enzymes is incorrect, DNA-dependent DNA polymerase, DNA-dependent RNA polymerase, RNA-dependent DNA polymerase, and RNA-dependent RNA polymerase, which of the four is not a correct enzyme? People, quickly.
The last one, RNA-dependent RNA polymerase.
Yes, that's not an enzyme. Okay. So DNA-dependent DNA polymerase is for replication. DNA-dependent RNA polymerase is for transcription. RNA-dependent DNA polymerase is for reverse transcription. And RNA-dependent RNA polymerase does not exist. Cool. So let's come to transcription then. Now, transcription, as we know, it's copying the information from a DNA to a RNA. So you can write down, it is the process of copying genetic information. The process of copying genetic information from one strand of the DNA. From one strand. Now pay attention to this word, highlight it, one strand. One strand of the DNA into RNA. Into RNA is transcription, okay? The principle here is also again same. You have to make complementary pairing and make a RNA. So this time the DNA will again open, but now we do not need to open the full DNA, right? Because transcription is for expressing a gene, right? Any information that you want to express in a cell, you need to make RNA of that gene. So gene is a part of DNA. It opens, RNA is formed, and that RNA comes out of nucleus and then gets translated inside the cytoplasm. Okay, I hope the picture is getting clearer to you as we are going forward. Everyone make sense?
Yes.
Yeah. So here also it is based on complementarity. The principle of complementarity governs transcription also. Write it down. The principle of complementarity. Just like it governs replication, the principle of complementarity, base pairing, simply base complementarity, governs transcription. Okay. But the difference here is you do not have thymine. So A makes a double pair with U. So if a DNA is open, so let me show you it like this. So let's say there was this DNA and it opened like this, not like this. I forgot that when it will be open. Okay, this is the DNA that opened. So the RNA that will be formed complementary to this strand. If on DNA there is adenine, the RNA will translate it as uracil, U. Okay, instead of T, there will be U. So RNAs have uracil and not thymine. Okay. Now, one question that comes in boards many a times is this. Write down this question. This is the last question we will discuss for today, but a very important one. I ask you to pay attention to this term that transcription is the process of copying genetic information from one strand of the DNA into RNA. Why not both the strands? Why just one strand? And which strand then? How to decide which strand? Is it always the same strand? Or do you think it does not matter? One time it can be one strand, the next time it could be the other strand. Can we copy genetic information from any strand or both strands, or very strictly it has to be one single strand all the time? What do you think? Quickly, since we have less time, I want some quick opinions on it. Think and then give an opinion. Hiram, what do you think? Why just one strand? Why can't we copy from both the strands? Mariam? Parker? By this time, you have to start thinking. And you all have the capability. Undoubtedly, you all can think. Think about it. Just think in your head. What will be the problem? Let me make a DNA for you. Let's say this is a DNA strand. Okay. This is the 3' to 5' strand, and this is a 5' to 3' strand. And this is the sequence A, E, C, G, A, T, E, C, G. Let's say this is the sequence of 3' to 5'. Now, to make a RNA, why can't I just choose any of the two strands? Does the question make sense to you?
So maybe because RNA synthesis.
Maybe because?
Maybe because RNA synthesis starts from 5' to 3' end.
But again, just like the DNA could have been formed, you are saying that DNA dependent RNA polymerase, this enzyme also have a direction, right? You are right. This also has a direction, which is 5' to 3'. So you are saying just because it has a direction. So what Iram is saying is half of the answer and is very important. She's saying that the enzyme DNA dependent RNA polymerase, which is doing transcription, I'm writing it as RNA pol. So this can only work just like its cousin brother, DNA dependent DNA polymerase, cousin sister. This can also work from 5' to 3' direction. It is also unidirectional, one direction. All these molecules are fan of one direction, just like us. So clearly, when it will start working, it will choose this strand, right, as a template, correct? That is what you are saying, you know?
Yes. Yes.
But you're missing one point. To do transcription, DNA opens up. When DNA opens up, why can't it choose this strand in this direction and start making, you know, because for transcription, we don't have this problem of transcribing the whole DNA length. That was the problem for replication. That's why there was Okazaki fragments being formed. Here, we don't care about transcribing the whole DNA length. Anyways, you will just open one gene segment and transcribe that one at a time. You don't have to transcribe all the genes in one go. So you don't have to worry about fragments. Anyways, you want fragments here. So once both the strands are open, you are free to choose any of the strand and still continue working in the direction you want. Don't you think so, Iram? It was a very good point brought up by you, but that is not a limitation here. Do you understand what I'm trying to say? Once both the strands are open, you are free to go in 5' to 3' direction on any strand. Okay. You understand how or not, Iram?
Yes, I got it.
You got it, right? The RNA polymerase can choose this strand also and go in this direction from A to G. It can choose this strand also and go in the opposite direction. It is limited by the direction, no? And here, there is no point or problem of making fragments because that's what we want. We just want one gene to be transcribed, not all. Okay, so I want all of you, Iram, Parker, and Mariam, to sleep over the problem, think about it. And in the next class, when I will teach you the machinery of transcription, before that, you will give me the answer. Write down the question. This question has come multiple times in boards also, that why both the strands of the DNA are not copied during transcription. What is the problem? What is the biological problem?
So,
They can, yes, Iram. Yeah.
I think I kind of got it.
You think you know the answer now? Okay, that's great. Give me the answer. There? Yes, I will repeat the question. The question is why both the strands of the DNA are not copied during transcription. The answer is not that they cannot be copied. They actually can be copied because I just told you that direction is not a problem here. It was a problem for replication, not for transcription. Okay. It can very well be done, but it is not done. Biology evolved in a way where it learned that if both are copied, there will be a problem. I am asking you why it is not done and what is the problem? Iram said she know the answer, but she's not telling. Iram.
So I'm confused.
Okay, that's great. A confused answer is a very good seed for germinating a very, very good tree out of your curiosity. So keep that confusion in head. As I told you, confusion, concept, and confidence all starts with CON. Okay. So if you're confused, then only you can be confident and build a concept later on. So keep this confusion, sleep over this confusion, try to find answers of this confusion. I'll just suggest you don't go directly on Google, because Google will just, like a slap, it gives an answer right on your face. But that does not evolve your thought process. Think over it, think the biology, and at the last, try to go to books and internet and try to understand the answer. Okay. In the next class, first you people will speak and tell me about the problem, and then I'll teach you the machinery.